Hello guys, welcome to this YouTube channel. A good day to everyone watching this video. In this video, I will be discussing the answer key for the biochemistry section of the GATE Excel 2019 paper. So let's get started. So the first question is, catalase is found exclusively in. So this is a basic cell biology question. The correct answer here is C, which is peroxisome. So as you may know, catalase is involved in protecting cells from reactive oxygen species or ROS, wherein it primarily converts hydrogen peroxide to water and hydrogen. So water and oxygen, sorry. So this is mainly involved in the decomposition of hydrogen peroxide and is involving in the protection of cells from ROS. So it is mainly found in peroxisomes. Moving on to the next question. RAG recombinase is responsible for the formation of specific immune receptors. So this process occurs in. So in the question itself, there is a slight key that there is a specific immune receptors. So which means it's most likely related to adaptive immune response. And also if you remember the VDJ recombination mechanism, RAG recombinase is involved there. So keeping these in mind, the correct answer here is T cells and B cells because these cells are involved in recognizing antigens specifically and eliciting an adaptive immune response. Okay, so this is a pretty simple question to solve if you know basic immunology concepts. Moving on to the next question. The example of substrate level phosphorylation in glycolysis is, so here the correct answer here is conversion of 1,3-bisphosphoglycerase to 3-phosphoglycerate. So the other three, if you look, conversion of glucose to glucose 6-phosphate or glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate or dihydroxyacetone phosphate to glyceraldehyde 3-phosphate, these are not substrate level phosphorylation. There are only two substrate level phosphorylation in glycolysis, and one of them is conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. So the correct answer here is option C. Okay, moving on to the next question. The dipeptide with least rotational barrier in the peptide bond is. So this question can be a little tricky because this is a chemistry question. However, if you look at the steric hindrance and the possibly the involvement of resonance structures, then the correct answer here is option B. If you look at all the other peptides, there is a proper CONH bond, which is a planar structure, and there is a kind of rotational barrier there. But however, if you look at this is not the canonical CONH bond that is usually seen in a peptide bond. So the correct answer here is this is the dipeptide, that is the option B, with the least rotational barrier. So it cannot be planar. However, it can have some rotation that's possible. Yes. Moving on to the next question. The light harvesting pigment not used by cyanobacteria for photosynthesis is. So here it's pretty easy. The correct answer here is rhodopsin. If you see the other three, that is phycobilin or phycoerythrobilin or phycocyanobilin. Phycocyanobilin already has the name cyano. So all these three are involved in light harvesting reactions for photosynthesis. However, rhodopsin is not found in cyanobacteria. Moving on to the next question. Slow intravenous infusion of ethanol is a therapy to treat methanol poisoning. So the underlying chemical reaction here is an example of the correct answer here is competitive inhibition. So this is usually used as a treatment, and if you know that this is an example of competitive inhibition enzyme reaction, then this is an easy question to solve. So this is like a basic, I would say, a general knowledge question here. Okay. Moving on to the next question. Nitric oxide synthase is responsible for the generation of nitric oxide, an important signaling molecule. The substrate for this enzyme is. So basically, I would request everybody watching this video to look at the nitric oxide associated signaling pathways. It's not just one, there are several signaling pathways involved. And the main substrate for this enzyme is actually arginine. And this is a pretty important enzyme, especially nitric oxide is involved in many signaling pathways and most of these reactions are, since it's an enzyme, since synthase is an enzyme, it requires a substrate, and it should also be resynthesized after the reaction is done. So in that case, the enzyme for the substrate for this enzyme is arginine. Yes. Moving on to the next question. Allergies are due to a hyperimmune response. Drugs given to counter allergies target. So this is a very simple question. So it's basic immunology question, and usually the allergies, the treatment given to allergies are usually antihistamines, which means they target histamines. So this is a very simple question to score. Moving on to the next question. The electrostatic interaction energy between a positively charged atom A and negatively charged atom B separated by 3 angstrom in water is minus 6 kJ per mole. Considering the relative permittivity of water to be 80, the electrostatic interaction energy in kJ per mole rounded off to 1 decimal place between atoms A and B in vacuum is. Okay, so this is a physics question, it's pretty surprising. Anyway, so for electrostatic interaction energy, the formula is, so let's first write for water. So E equal to K Q1 Q2 by R. So basically electrostatic force is K Q1 Q2 by R square. So for energy, you multiply it by the distance between them. So that leaves you with the expression K Q1 Q2 by R. So this is equal to minus 6 kilojoule per mole. This is for water. And if you say K is equal to the dielectric constant, which is 1 by 4 pi epsilon water, which is epsilon is the permittivity. And if you look at the vacuum, let's write the expression for vacuum. E vacuum is equal to, let's call it K prime. And the charges remain the same. K Q1 Q2 by R distance is also the same. So that is going to be what that we need to find it out. And for K prime, we know that it is 1 by 4 pi epsilon naught. Okay, the permittivity in free space where epsilon naught is. And relative permittivity, which is ER, is equal to 80. That is given in the question, which is basically the dielectric constant. So that is equal to E water by epsilon water by epsilon naught. So now if you divide E water by E vacuum, so you get E water by E vacuum. So that is going to be minus 6. Let's call E vacuum as X just to keep it simple, is equal to epsilon naught by epsilon. So epsilon water. Why? Because it's in the denominator, it goes up. So epsilon naught by epsilon water. Okay, so this is equal to 1 by 80 because epsilon water by epsilon naught is equal to 80. So epsilon naught by epsilon water is equal to 1 by 80. So now X is equal to minus 6 into 80, which is minus 480 kilojoule per mole. I hope this is pretty simple to solve. If you know the basic physics formula here, like the first chapter of electrostatics in your CBSE paper, then this is a pretty simple question to solve as long as you don't get confused between relative permittivity and dielectric constants. So that is the key here. Anyways, moving on to the next question. You are given a 0.1 molar solution of glucose, which is the stock solution, and the stock solution required to make 0.5 ml of 0.005 molar glucose solution rounded off to 3 decimal places in ml is what? So this is basic dilution formula question, which is C1V1 equal to C2V2. So let's call C1 as the stock concentration and this V1 as the volume of the stock concentration that is required. And 0.1 into V1 equal to 0.5 into 0.005, right? So this is C2, this is V2. Okay, and V1 we need to find, and C1 is this for the stock. So now V1 equal to 0.5 into 0.005 divided by 0.1. That will give you 5 into 0.005, which is 0.025 ml. Since everything is in ml and they also ask the answer in ml, it's very important to check the units in which the data is given and the way in which they also ask in the answer. So the correct answer here is 0.025 ml. So this is pretty simple. Moving on to the next question. A mixture of the following purified proteins, IgG, IgM, and Fab fragments of immunoglobulins is separated using gel filtration chromatography. The order of elution of these proteins is from first to last. So basically, from first to last is asked, which means from the large protein to the small protein. So we know that IgM is usually a pentamer, so it is likely to be more bulky. This is followed by IgG, and this is next followed by Fab, which is obtained using the proteolytic cleavage. So the correct answer here is option B, that is IgM, IgG, and Fab fragment, from large one to the smaller one. Anyways, this is pretty simple. Moving on to the next question. The ascending order of half-life for the radioactive isotopes is what? So this is a pretty, I would say, a factual based question, if you can remember this, because I wouldn't give so much weightage while preparing for these type of questions. However, for the sake of discussing this paper, I would say the correct answer here is option B. However, one way by which I would actually guess this, guess the answer for this question by deductive reasoning, if I want to take a risk in the paper, would be that I125 and 32P are pretty unstable compared to H3 and C14. So if I can look at the options where, the half-life is really less for these two, then I would definitely choose this option. However, if it is really confusing and then you cannot remember the exact values, I prefer skipping this question instead of scoring a negative marking, and then go on to the other ones. And if possible, if you can recollect it through deductive reasoning, then yes, go ahead in the last minute. Yes. Moving on to the next question. The enzyme not involved in the oxidation of the following molecule is. So the correct answer here is carboxylase. Of course, it's involved in the carboxylation reaction, which is removal of carbon dioxide, which is not an oxidation process. However, isomerase, dehydrogenase, and hydratase can lead to oxidation of the following molecule given in the picture. So the correct answer here is carboxylase. This, I would say, instead of knowing the exact mechanism by which each of these enzymes involve, if you can deduce this based on the names here like isomerase, carboxylase, or dehydrogenase, or hydratase, then it's pretty easy to guess the correct answer for this question. Moving on to the next question. DPN1 is used to digest the PCR product during site-directed mutagenesis because. So DPN1 is an enzyme and it is involved in the digestion of only methylated DNA. So this is like a basic fact that is known, that you would have read during SDM or site-directed mutagenesis, and especially when you have to do a PCR product and then digest the PCR product that has this methylated DNA. Anyways, moving on to the next question. Which one of the following is an incorrect biomolecule modification pair? So here, the correct answer here is lipid palmitoylation. First of all, this question can be very easy to get lost in, and many people, including me, if I just see the question at the first time, if I'm not really aware of what is happening to what type of biomolecules, then people can easily choose protein methylation, which is very easy to get lost in. However, palmitoylation occurs when lipid molecules are attached to other biomolecules. However, palmitoylation itself doesn't happen on lipid molecules or lipid biomolecules. So the correct answer here is lipid palmitoylation, because on lipids you don't see this occurring. However, palmitoylation occurs on other biomolecules like proteins. So the correct answer here is lipid palmitoylation, and I request everybody to be really careful while choosing the answer for questions like these, where words can be misleading. Anyways, moving on to the next question. The crystal structure of a peptide has an ordered structural repeat of amino acids with a distance of approximately 6.5 angstrom between the alternate C-alpha atoms. Which one of the following pair of dihedral angles accurately represents the peptide structure? So if you are a really good structural biologist and you can remember the the basic idea of Ramachandran plot. So this question is based on Ramachandran plot, and here one clue that they've given is the distance is roughly 6.5 angstrom between the alternate C-alpha atoms. So this likely indicates a beta sheet. And if you can remember the Ramachandran plot, the beta sheet is in the upper left quadrant, and that makes you choose the answer as 5 roughly as minus 120 degree and psi as roughly plus 120 degree. So the correct answer here is option B. Moving on to the next question. Absence of detectable protein expression upon blunt-ended mutation-free cloning of an E. coli gene with its own promoter in E. coli cells can be due to cloning occurred in reverse orientation. No, this cannot be the case because nothing is mentioned like that. Cloning occurred out of frame. That is also not the case because it's under the influence of the promoter. And there's a codon bias. No. And last one is rapid degradation of the expressed protein. So this is a possibility because the protein is overconsumed or rapidly degraded. So either of the options could be true. And here the correct answer here is rapid degradation of the expressed protein because it's an E. coli protein which is mutation free and is expressed in E. coli itself with its own promoter. So this is rightly the reason for the question. Anyways, moving on to the next question. The C-terminal carboxyl group and the N-terminal amino group in amino acids have a dissociation constant pKa of 2.2 and 9.2 respectively. Okay, and the pKa of side chain carboxyl group in glutamic acid is 4.2 and the side chain amino group in lysine is 10.2. The difference in isoelectric point Pi of lysine and glutamic acid rounded off to 2 decimal places is. So first let's write for glutamic acid. This is a very simple question provided you write it correctly and don't mess up with the correct pKa values. So for glutamic acid, it can go from glutamic acid plus 1 to glutamic acid 0, glutamic acid minus 1 since it's a negatively charged amino acid. It can also have glutamic acid minus 2. So now write everything in ascending order. pK1, pK2, and pK3. So here pK1 is 2.2, here pK2 is 4.2, and pK3 is 9.2. So if you have listened to my previous videos or seen my previous videos, you should take the one where this neutral charge is involved with plus 1 and minus 1 on the other side. So that gives you the isoelectric point here because it's the pH at which the amino acid is electrically neutral. So here the Pi would be equal to pK1 plus pK2 divided by 2, which is 2.2 plus 4.2 divided by 2, which is 6.4 divided by 2, which is 3.2. So let's keep it here. Now let's go with histidine. So for histidine, we know it's a positively charged amino acid, so it can go up to histidine plus 2 in equilibrium with histidine plus 1, in equilibrium with histidine with electrically neutral charge of 0, and in equilibrium with histidine minus 1. That's the maximum negative charge it can go. And here if you have pK1, pK2 equal to and pK3 equal to, write it in ascending order. That will be 2.2, this will be 9.2, and this will be 10.2. And similarly you should take this one, so which is Pi is equal to pK2 plus pK3 divided by 2, which is 9.2 plus 10.2 divided by 2, which is 19.4 divided by 2, which is 9.7. Now they ask the difference between the isoelectric point of lysine and okay, this is not actually histidine, this is actually lysine. Anyways, not a big deal. Anyways, lysine is also a positively charged amino acid. And let me just change it here in the question just to make it easy or less confusing. Okay. So here they ask the difference between the isoelectric point of lysine and glutamic acid. So you just have to take the difference here. So Pi difference, let me call it this way, that will be equal to 9.7 minus 3.2, that is 6.5. So the correct answer here is 6.5. I hope this is a very simple question to solve. Anyways, moving on to the next question. X different size DNA fragments can be observed upon incomplete ECO-R1 digestion of identical DNA molecules with two ECO-R1 sites as shown below. The maximum value of X is, so here this is an incomplete digestion using eco-R1 restriction enzyme. So either only one site is being cleaved off. So either only at one site the cleavage occurs, or there is no cleavage at all. So if at one site a cleavage occurs, then this yields 500 base pair DNA fragment and 1000 base pair DNA fragment. And if there's no cleavage occurring, if basically the concentration of the restriction enzyme is too low, then even it might not be able to digest all the DNA fragments. So in that case, the DNA fragments would be undigested and remain as 1500 itself. That is 500, 500, and 500. So in that case, you have three types of DNA fragments of different lengths being generated here. So the maximum value of X is 3 because here it's incomplete digestion. It's a very simple question if you know basic genetic engineering. Moving on to the next question, or the last question. An uncharged protein P has an aspartic acid in position 23 with a molecular weight of 6501 Dalton as determined by mass spectrometry. And the uncharged mutant of this protein, that is P prime, contains a single amino acid substitution with asparagine at position 23. The molecular weight of P prime as determined by mass spectrometry rounded off to 1 decimal place is what Dalton. So first of all, you should know the structure of aspartic acid. I will just write the side chain here, which is CH2COOH. And for asparagine, it is CH2CONH2, right? So here, the difference is it's a carboxylic group, and here it's an amide group. So here, if you look at if you just take the molecular weight of OH, it's going to be 16 plus 1, which is 17. Atomic mass units, and if you take this, it's going to be 14 plus 2, which is 16. So if you look at the difference, it's just plus 1, extra 1 AMU for aspartic acid. So now if there's going to be a mutation with asparagine, then the molecular weight is going to decrease by 1 Dalton, which is 6501 minus 1, which is 6500 Dalton or 6.5 kilodalton. However, they have asked the answer in Dalton, so the correct answer here is 6500. So this is a very simple question to solve if you know basic atomic weights of the elements that we have learned during our 10th or 11th grades, and then if you can know that where the substitution occurs and where the mutation occurs, if it's at the same position and also the same protein, then it's going to be a very simple question to solve. So with this, I'm completing my discussion with the GATE 2019 Excel paper biochemistry section. I hope you found this video interesting, and I hope you found this video useful also for your upcoming GATE exam preparations. I wish you all the best and stay tuned for the upcoming videos. So have a nice day. Goodbye.